Awesome.
All right,
All right.
wonderful. All right, well, thank you very much for having us today. We are Renegade Research, and we'll be focusing a good portion of the time on the Remission Biome Project, as um, Mr. Esteban mentioned. Next slide, please. So on the agenda, we're going to be going over a little disclaimer, talking about us, our organization, Renegade Research, and our team, MECFS, the Remission Biome Project, from history to lessons learned. And then we're going to share a little bit of how to get involved if you are interested. Next slide. All right, so disclaimer, the content presented in this session is for informational purposes only and is not intended to be a substitute for medical advice, diagnosis, treatment, or treatment suggestions. Always recommend seeking the advice of a qualified physician or other qualified healthcare provider with any questions regarding a medical condition. Next slide. So the three of us here today, I am Isabel. I'm the Remission Biome Project Director and the CEO of Renegade Research. Some of my passions are research and empowering patients. I've had MECFS for 43 years. I've been married for 30 years and have 3 adult children. I am obsessed with community living and live it every day. And I love foraging, gardening, hiking when MECFS allows me to do so, and all things homesteading. And Kat?
So I am Kat Boniface. I'm a former horse trainer. I used to say once and former, but at this point, I think I need to step back from that a bit because, of course, MECFS is quite limiting and quite unpredictable. But despite that, just like Isabel, I have managed to do quite a lot in the meantime. So I did have a horse fall on me, had knocked me out. I had a TBI in 2008 and had fairly immediate changes from that I now recognize in retrospect as MECFS. But I did continue on through my undergrad, through my MA, and through most of my PhD, focusing on the history of animals, animal science, animal technology, particularly genetics and nutrition, sort of bringing my background from my farm work, from my veterinary assistant work to my historical research, and now to my personal research as I shifted to looking into my own health before finding Renegade Research. Um, I am in Southern California, technically still associated with the University of California, Riverside, although that is currently on hiatus. But I am currently quite hopeful, as we've managed to make quite a lot of progress, that I will be able to return to that as well. And Tess.
Hi, I'm Tess. I'm founder of Remission Biome and Renegade Research. And like David mentioned, my background is in something very different. I used to be obsessed with space, and so for a long time, I studied space science and did aerospace engineering. But I got MECFS my senior year of college, like the summer right before I was working at NASA JPL, and I got valley fever, which is a fungal infection in California, and never recovered from that. And so yeah, I was sick through most of my career in aerospace, and then from that got me to where I am now, working on these conditions and using my research background. Even though it's in a very different field, it all still kind of applies the way you think about science and research and systems, complex systems, which we'll talk a bit about today.
All right, so the way we like to talk about us is we're catalyzing change with patient and caregiver-led research. Next slide. Okay, so who are we? Renegade Research is an organization, is nonprofit, 501c3. We focus on complex multisystem chronic conditions, such as MECFS and long COVID. We do our research in a very collaborative and decentralized way, and we also focus on education and practical advocacy. So we are building a network designed to improve access to care, access to research on all levels. Remission Biome is our largest project, but we also have 10 additional projects that are in various phases of development, including a partnership with OMF. Next slide. So we are a great team of mostly volunteers who work on different aspects of the organization, from data analytics to directly in doing the research to different things that are needed to run the organization that we're running. And everyone is incredibly invested, incredibly collaborative and cooperative. And every single one of us has some experience with ME/CFS. About 96% of our volunteers either have a condition or are a direct caregiver for somebody with the condition. So everyone is incredibly invested in seeing solutions come through. Next slide. So ME/CFS, as you guys know, is a multi-system complex chronic condition. One of the hallmarks of ME/CFS is post-exertional malaise, which is an exacerbation of symptoms or new symptoms that are not normally part of the presentation of a particular patient. So post-exertional malaise differentiates it from many other conditions that share similar symptomatology. But some of the most common symptoms are fatigue, which we always say is a very inadequate way to describe what we go through, right? Everybody has fatigue. If you work a full day, if you work 2 jobs, if you're, out running around the entire weekend, if you're out late, fatigue is a normal part of what you experience afterwards from doing a lot. But the fatigue that ME/CFS patients experience is more like a total battery drainage. And it is often very disproportionate to the activity that it's done. On the more severe end of the spectrum, patients can go into post-exertional malaise simply from getting out of bed and going to the bathroom to brush their teeth, or other people may experience it from having to prepare a meal for themselves, while others can work a full-time job but need to spend the rest of the day or the rest of the weekend resting in order to be able to do it all over again. Cognitive dysfunction is also a common symptom, and orthostatic intolerance, dysautonomia presentation, is also a very regular comorbidity. There's a number of symptoms. We won't list them all. And again, there's overlap with other conditions, but post-exertional malaise is, one of the cardinal distinctions of this condition. Next slide, please. So what are the challenges to treatment that we face? So first of all, the condition is undertaught in the clinical setting, right? So we have few knowledgeable doctors available, and the ones that are available and have the expertise and have been taking care of patients for a while are oversaturated because, everybody wants to go to them. And then those treatments that they provide are not always covered by insurance, if patients even have the access to insurance in many countries. We have siloed medical systems as a whole, not just in ME/CFS, but in this instance, patients are bounced from specialist to specialist because, again, is not something where you can pinpoint what the issue is and treat that issue. Is a multi-system condition that affects many, many parts. So all the systems affected don't fit neatly into a box. And there are a few clinicians with the expertise to address all of the symptoms and know how they all fit together. Patients have energy limitations. So this is difficult because, just accessing care can be a barrier. It presents in a heterogeneous way. We saw the list of symptoms, but not all patients experience all symptoms, and not all patients experience the same symptoms in the same way or with the same regularity. And then the heterogeneity of the response to off-label treatments, right? We often say that a treatment that works wonders for one patient doesn't do anything for another, and yet makes another one worse. So fitting the system and working systematically with the patient, it's critically important. Patients experience sensory and environmental risks. So somebody who is, for example, able to go to an office and work, may not be able to do so, not because of the energy limitations, but because the office is loud, bright, and they have to be upright the entire time. So their threshold for going into PEM is decreased, not just by the energy expenditure, but by other factors as well. And then the treatment is rarely straightforward, right? It's similar to cancer is the closest analogy. It's long, complex, needs to be customized to the patient, and it requires a multitude of supportive therapies in addition to the central treatment. So protocols are designed to address those individualities and have the flexibility to work with the patient, but also have replicable data at a cohort level. Next slide, please. So our collaborative research, again, we're patient-led and well-suited to be decentralized. Good standard of care for ME/CFS at a scale requires decentralization. So that's what we're working towards. ME/CFS patients, by necessity, are very well educated on their disease. When you speak to patients, they often know more about the condition, treatments, and the systems than a lot of clinicians, unfortunately. The emergence of long COVID has increased the need for resources. And we have now 5 years into the COVID pandemic, about 50% of long COVID patients meet the criteria for ME/CFS. So the need has only increased exponentially. And when we look at future pandemic possibilities, if we don't get a handle on this, the exponential growth is going to continue. And our organization is working towards seeing exponential decay of the suffering of ME/CFS patients. So because of all of this, there are many patient-led research projects that are now underway. Researchers and clinicians also become patients, not just in long COVID, but previously with ME/CFS. And Renegade Research is fostering a collaborative environment where everyone can get involved. So we'd like to collab and conquer. Next slide, please.
Okay, so now we'll dive into our largest project, Remission Biome. Okay, so this project actually started on Twitter. This is how it all came together. There were a bunch of patients. There's a large community of ME/CFS and long COVID patients on Twitter. And we started talking about the fact that many of us had dramatic and complete remission of ME/CFS after taking antibiotics. This happened to me in 2009. I went from nearly bedbound to jumping out of bed within 2 days of starting a course of antibiotics. All symptoms gone, tons of energy, and it was a wild experience, and I had no idea that this had happened to other people until getting on Twitter. And then ended up finding out that Isabel and Kat both also had this happen before. So we'll all kind of tell a little bit about our experiences throughout this. But yeah, so there was all these anecdotes. We started discussing hypotheses, like how could this actually happen? We don't understand, all of the pathophysiology of ME/CFS. So it was like, this is a clue that if we're able to turn it off like this, and we can study this, we can understand more about how this could happen, and that'll tell us about ME/CFS. So we decided to try to recreate the transient remission events on purpose, taking antibiotics and seeing if we could actually trigger one of these events. And the Twitter community just got really excited about this. We ended up getting a ton of momentum behind this, and people started crowdfunding. So the idea was, all right, let's do a self-experiment, but physician supervised in case anything happens, measure a lot of biomarkers before, during, and after, if one of these remission event happens, do most of the sample collection at home. So with at-home test kits that you can then mail in, which allows you to, sample during specific times. If you were in a traditional clinical trial, it would be really hard, if somebody had a remission event, to be able to run into the lab and get there on time, get all the samples that you needed. So this was a key part of what we do, is to keep things decentralized, people collect at home. And then doing things like daily symptom tracking to keep track over time throughout the experiment. And I mentioned the community crowdfunded for us. So that's how the experiment grew. So it started in the fall of 2022 was when these discussions were happening on Twitter. And the pilot was planned to happen in the beginning of 2023. And we actually got a cryptocurrency grant to be able to cover a lot of the testing for that pilot study. So you can already see we are very non-traditional. We decided to do this in a different way. And so we had the crowdfunding, we had a cryptocurrency grant, and then we worked with sponsors of different products to be able to get free products or discounts to be able to do the experiment. And you can see it just continue to grow throughout this presentation. We'll talk a bit more about how the project expanded and what it is now, what we have learned. But we did the pilot study in March and April 2023. We started getting ready for it in February. And then there was this phase we call the 1.5 phase, which was in between the pilot study and our current 50-person cohort. And then we call that the phase 1.5. And then towards the fall of 2023, we then turned this into a larger project, which we call the Renegade 50, which is 50 people from all around the world working with their own healthcare providers to do the Remission Biome protocol. Okay, so mentioned hypotheses that we were discussing on Twitter. And I'm sure you all, when you're learning about the research and the science behind ME/CFS, you realize that it's complicated. This isn't just a simple condition where you have one biomarker, you can take one medication, and you are able to, treat the symptoms or, I don't want to say cure the condition, but ME/CFS is multisystem. There's a lot of stuff going on. We don't really have good treatments. So when we started thinking about hypotheses of what might have happened, it quickly got complicated, thinking about many different possibilities. And so some of the possibilities that we thought about was like, is there something happening in the microbiome? Are you killing off some bacteria that's producing some metabolite that is causing ME/CFS symptoms? Or are you opening a niche in the microbiome for a good bacteria to flourish? And so those were, two possibilities. We also thought a possibility what if this doesn't even have anything to do with the microbiome? What if the antibiotics itself are going into the system? And antibiotics some of them are anti-inflammatory. So there were some hypotheses that, maybe this is directly affecting the immune system. So I won't talk through this whole diagram. I just wanted to show it, to talk again about how complex this condition is and how difficult it is to study. And I want to take this opportunity to let you all know that, if anybody wants a major mystery to solve, a problem that is affecting many people that does not have easy answers, this is a good challenge if anybody wants to go into studying ME/CFS. So I mentioned, we're kind of thinking about what's happening in the gut. How does that then affect what's happening in the body, like in the immune system and in the brain? And how could that lead to these sudden dramatic remissions? And I will pass it off to Kat to talk a little bit in detail about some of the ideas and additional hypotheses in her experience.
So anytime we build a hypothesis, we then, of course, have to test it. One of the original keystone pieces, even going into the pilot trial, was to address neuroinflammation. Unsurprisingly, this, across ME/CFS, neuroinflammation is incredibly common. And even if it wasn't a direct driver, which, it likely is, at least in part, maintaining the system, it's certainly a barrier to recovery. So that was one of the first things that we needed to address, because there are so many different processes that just simple inflammation can drive. And if you don't remove that inflammation, then there is no way forward. No amount of other processes can fix what is being blocked by that inflammation. But we're going to go a little bit more into detail. Let's go ahead and go into our next slide. So in terms of, is it the antibiotic action of the antibiotic, or is it the non-antibiotic action of the antibiotic? Chances are it really is both. But it was very clear that the antibiotic action was not what was flipping the switch. And this is because of the simple timing. So from patient observation, the timing of remission events really just did not fit with it being simply removing pathogens or resetting the microbiome. So there had to be something else going on. But of course, we have to think systemically, we have to think long-term. Those effects on the microbiome become important later. But in terms of identifying why these initial remission events happened, there had to be something else going on. So this is where we came to a theory regarding glutamate. So I was one of the original pilot study participants. I had been drawn to Remission Biome because I had put together my own protocol previously that was shockingly similar and had worked. I had about a 7-month remission from it. And that was, ending, and I was declining again. And then I found Remission Biome, and it was fantastic because I was able to trigger the exact same event again, partially using amoxiclav, and it was much faster this time. And that speed was really, really important. So we started looking at, okay, well, where are the commonalities? What is possible that amoxiclav could be doing that could flip that switch? So many of the supportive treatments for ME/CFS are aimed at reducing glutamate excitotoxicity, so excess glutamate signal that can then eventually cause additional neuroinflammation and even cell death, which can, create a vicious cycle. Now, in collecting data across Twitter and eventually beyond, there was a distinct difference in response to amoxiclav, which is what we had decided to use, and amoxicillin itself. So there had to be something with the clavulanate. So this is what I started digging into next. And the primary difference between the two of them was its action on one particular, supposedly small, minor glutamate transporter. So not looking at the signal, but instead at the metabolism. So this was the case of patient observation plus this community of researchers coming together to make this breakthrough. And now testing that hypothesis, looking at how other treatments work, how people respond, has led to more targeted treatments for individuals. So looking at this, large, very heterogeneous population and trying to figure out who is actually going to respond to different types of treatments. Even within those, treatments for excitotoxicity, there are a lot of differences, even just within that one category. And as we can start to really, really hone in on these really specific mechanisms, we can identify who's going to respond better and who might respond poorly to a particular treatment. And this is a little bit more recent, and we're still investigating, still need to do a lot more research on this, but digging further into this has also potentially illuminated a neurological switch for PEM itself. So for not just the remission events, but also the sort of inverse of the remission events, which is when PEM itself is triggered. All right, next slide.
All right, Isabel's up again.
All right, so we're going to go into the Remission Biome Protocol. Next slide. Okay, so the protocol originally consisted of 3 phases, which is phases 3, 4, and 5, as you see over here. But as we got going into the project, there were a lot of things that we were able to learn, including some challenges that patients present with in terms of comorbidities, co-infections, reactivations, and so forth, that clearly needed to be addressed to stabilize participants to go into what we now call the core of the protocol. So that was what led to this expansion. So first, in stage 1, it's a preparatory phase that stabilizes those, other issues that are ongoing, and also where a lot of the testing is done. And we're going to look at some of that in a moment. So once that very individualized approach is done in that phase 1, then we move into a 2 to 4 week period of just stabilization in terms of no additions, no subtraction of anything else during that phase. And then phase 3 of the protocol is a gut-brain preparation, where a series of interventional supplements are designed to address inflammation, to address the gut microbiome, and to address neuroinflammation in preparation for the antibiotic phase, which is phase 4. So that phase is 14 days long, and that's where we expect to see in some participants transient remission events. And these remission events can last anywhere from minutes to days, and in some cases, even months. But one other important aspect of this is that baseline increases are also seen as a result afterwards. After the phase 4 with antibiotics, we move into phase 5, which is a stabilization of the microbiome again, and continuing to reseed and to work on brain-gut axis and continuing to address neuroinflammation. And then stage 6 is more stabilization, making sure that the trajectory continues to be upward, and that many of the issues that we saw in stage 1 are now addressed, and expecting an increase in baseline function as well at this stage. Next slide. So some of the things that we do during all of the phases, right? Participants report data in the terms of symptoms, and we also collect objective data. So participants log their medications, supplements, probiotics, not just what they're taking as part of the protocol, but what they're taking in general. So we can look for signals, that may be an impediment that may have aided into the results as well. And we do this through an app called Wearables. So they also have data that is related to mood. And in this case, mood for us means your overall well-being, right? Because of the heterogeneity of the presentation of the condition, there are many symptoms that are part of the day-to-day. There's also function that is not always coupled with those symptoms. So that mood is on a scale of 1 to 10, and it gives us a more generalized overview on how a participant is doing on a particular day, and more importantly, what the trends are. We also do brain check. We look at Bell scores, FUNCAP. Those are all tracked. Those are validated questionnaire that are tracked throughout the participation. And then we take information from wearables, Oura Ring, Garmin, Apple Watch, and look at particular stats such as resting heart rate, HRV, sleep, which is also, very disrupted in ME/CFS, and looking at how that changes and what the sleep architecture is throughout participation. And for particular labs and tests, things like Biome Site, which tells us, what the microbiome composition is before, during, and after, organic acid test, and a bunch of other standard laboratory tests as well. We look at genetics, and again, looking at, what are possible co-infections or impediments as well. What are there mold infections? Does somebody have Lyme going on? And all of that together tells a much bigger story of what's going on within the individual. Next slide.
All right, now we'll talk a bit about what we've learned throughout this process. So the first thing I mentioned when we first got started, the very first question was just, can we purposely trigger these remission events? And I honestly did not think we were going to. I thought this was just a rare thing that happens, that it wasn't actually, we weren't going to actually be able to do it. But I was like, well, let's try, let's make some measurements. So we checked off that first goal. Yes, we can purposely trigger them. It doesn't happen every time, but we can. So for the pilot results, this was, I mentioned, in March of 2023, we did a ton of testing. And this is just one example that this was single cell immune profiling. So this is my own personal data, and this was sampling I did at home with a device called the Tap2 that you connect to your arm, and it like gets your own blood sample, and then I had to ship it back to this company in San Francisco quick enough that they could, the sample would be in good condition to analyze. And they ended up finding some really interesting results in my data. So these are looking at, immune cells. So these are classical monocytes. And in the purplish bluish color there, those are samples that were taken in the months before I took the antibiotics. And red is during the antibiotics. And then green is for a few months afterwards. And this similar trend of this gene expression going down in the monocytes during and after was actually found in looking at 21 different genes. And so this is an interesting clue that we'll follow up on. Something did happen in the immune system. And we can't say, did this cause the remission events? Was this just due to antibiotics? There's still a lot we don't know, but that's one of the interesting things about what we're doing with Renegade Research is we're looking for clues. We're looking for signals that then can be followed up with larger studies and more, clinical trials, depending on what we find. So, there are patient anecdotes in the community. We study them in a systematic way. We bring other patients together to see if we can recreate the results, and we spread the information then to academia and the community so that other people can follow up on it. And if you want to learn more about this particular results, we actually have a full research roundtable on our YouTube channel. So the YouTube is Remission Biome. And the IMU company that ran these samples, you can see it's an hour-and-a-half presentation. They talked in detail about what we saw and what this means. And there's also a blog about it. So we can put some of these links in the chat. All right. Another interesting finding. This is also my data. And we had a preliminary results meeting discussing some of the pilot results and some of what we've seen so far with the Renegade 50 study. And if you want to see that full meeting, is also recorded and on our YouTube channel. But this is just another, again, like interesting result, looking at all of the different microbiome samples that we've had so far. This one stood out here with all of this teal lactobacillus. And it so happened this was during a remission event. So again, interesting clue here, something to follow up on. All right, in that same meeting, I mentioned that we talked a little bit about some initial preliminary results from the Renegade 50. So a lot of participants still have to go through the process, but we have a ton of data collected already. So this was on the left, just one example of the sort of analysis that we can do with the data we're collecting. So this was using that Wearables symptom tracking data that Isabel mentioned. And this was looking for what symptoms tend to cluster together. And this might be able to tell us about the different subtypes of ME/CFS. There were a lot more results presented that day. So if you want to hear more, you can see some of the examples here on the right side. So some talk about some of the wearable data and some of the biome site results. Kat talked about genetic patterns. And then I talked a bit about what we've learned so far of how people do, like how many people have their remission events and how people do afterwards. All right.
All right. So one of the benefits and also sometimes frustrating things about doing a large, fully decentralized, not beholden to sort of traditional frameworks research project is there are just a lot of variables. And we want to be able to reach everyone. So we can't just say that, the people who don't really fit within this model aren't going to be part of this because, if they have ME/CFS, they should be able to access the same treatments. But the question is how do we make that safe and effective? And these are just a few of the things that we have come across. So stabilizing comorbidity sounds simple, sounds obvious, right? But the problem is how do we actually decide what's a comorbidity and what's actually a driver of disease? Because often these things overlap. As for example, Tess had a fungal infection. Now, there can be an acute infection that is actually a trigger or a driver of disease, or there can be a chronic underlying infection that isn't actually the trigger, but still maintains that sort of vicious downward cycle that keeps us in this hypometabolic state. Considering off-label medication effects, most of us, even under direct prescriptions from our doctors, are on a lot of different medications, many of which have interactions that aren't usually on label, that most medical professionals aren't going to be familiar with. Many will have distinct familiarity with one or two types of medications and all of their off-label effects, but not necessarily with the whole pharmacopoeia. So figuring out what some of those common ones are that affect large portions of our population. Just as an example, something very, very simple, Prozac. You wouldn't expect Prozac to have an enormous effect on hormones, but it does. It is an aromatase inducer, so it will increase estrogen, which for some people is a great thing, and for some people is a disaster. So just keeping track of those off-label things, even if they aren't ones that would be immediately detrimental to, an otherwise healthy person in our population, are going to be important. Mechanical factors, of course, and we'll get into a little bit more of this in a moment. That environmental and genetic factors greatly outweigh the actual trigger event in terms of symptom profile and in terms of response to treatment. So as an example, Tess had a fungal infection. Isabel had a viral infection. I had a TBI. You would think these would be three different diseases, but we all ended up in the same place. But people who have particular genetic factors or particular current environmental factors often have significantly more in common with each other than they do with people who had the same trigger event. And then that systems approach that we've mentioned throughout is most critical, not for the remission event itself, but for the durability. Treatment responses also change as our baselines change, whether it's, because we've stabilized comorbidities, because we've removed co-infections, or simply because our metabolic and immune state has changed. So the treatment also needs to change. And this is something that our medical system isn't really used to doing, that we have to constantly have this iterative treatment, even within a single individual. And then, of course, connecting all of the existing hypotheses regarding ME/CFS into a single model. And this is where, again, I come back to glutamate, which is, my personal research area. And then finally, that community and guidance is an absolutely key factor. We cannot underestimate the value of this. Of course, in terms of morale, that's super important. But even just in terms of the information itself, there are things that, we never would have thought were actually related symptoms until we start talking to each other. When I start having, a particular set of reactions, my right toe will itch. Totally random, right? something I never even mentioned to doctors until years in, until I was like, you know what, look, this absolutely clusters to my other symptoms. I do not understand how, right? And then I started talking to other patients, and there was a pattern. Every single one of us that had this sort of really weird, isolated, like single portion of our body itch that would come on at a particular, it was a gluten sensitivity. You know, not something that we would have thought to put together had it not been for community. So we cannot underestimate the value of that, not just to the individual in terms of the support needed, but even in terms of the research and furthering the science itself. All right, so for a couple of examples, we'll go to the next slide. So as I mentioned, those environmental and genetic factors really, really do far outweigh the trigger event. Just a couple of examples here. Ochratoxin is a particular type of mycotoxin. I've been seeing that very, very commonly in a certain set of symptoms. And because it affects a lot of the same systems that we're looking at, including glutamate, it also is a major barrier to treatment. Mechanical factors are one of the things that often do not remit. So for example, I probably would not have ever gotten to the point of actually getting at least a tentative diagnosis for thoracic outlet syndrome because it was completely hidden by the rest of my symptoms. But every time I have had a remission event, that set of symptoms remains. So there are certain things that do remain despite everything else, and those tend to be the ones that are mechanical. And again, as mentioned, that systems approach being really, really critical for the durability. Transient remissions are absolutely common from just a single intervention. You know, nearly every person we have talked to with ME/CFS or with long COVID has had some form of transient remission, but it might be hours, days, weeks at best, unless there is that systemic support. Whereas those that have that systems approach, I've been seeing a pattern of about 7 months seems to be the most common marker for how long those remissions last before there needs to be another major intervention. And that's an enormous difference in terms of the actual quality of life for the patient. And of course, it's a huge clue for us as we're researching into, well, what actually causes all of this. All right, let's go on to the next slide. I think I will pass it off.
All right, so a lot of information that we have shared with you today. We have a lot of our roundtables and webinars that go into a lot of detail posted on the Remission Biome YouTube channel. So I encourage you to go there and look at the areas that may interest you to learn about more. If you want to stay abreast of the events that are coming up, and these events are, again, roundtables and webinars where we bring either researchers or clinicians to share emerging research, their hypotheses, or in the case of clinicians, their experience with treating patients. So you can go to our website, renegade-research.org, and go to the events page, and there you can sign up to receive our newsletter. Next slide. You can also follow us on social media at Renegade Res, Remission Biome, and those are our personal Twitter X handles. And if you would like to stay in touch with the community, if you would like to participate in what we do, we welcome volunteers, and we'll post a volunteer application in the comments as well. Next slide.
I think that's it.
I think that's it. Okay.